Right, Sds-Page is a technique that we use in the biochem laboratory to separate proteins based on their sizes, and it is almost always used to assess the purity of our protein sample. And so remember that charged particles, of course, move in an electric field, and we're going to make those particles travel through a gel in this case. And so if that's true, then if everything else is equal, and we'll talk about what that means, that small particles are going to go farther in a given amount of time than larger particles. And just to reiterate what we mean by all of this, here is sort of what a Sds-Page gel apparatus would look like. Ours is a little bit different than this, but the same idea that we're going to run this vertically. We're going to put sample in the top of one of these wells, and we will apply a voltage once we do that. So we've got a negative terminal here and a positive terminal down there. We'll see we're going to have negatively charged proteins, and they're all going to then migrate this way. And so if we have a mixture of different size proteins, and that will mean that the larger proteins will get more caught up in the mesh, and the smaller ones can cruise on through. And so we will see that smaller proteins migrate farther and will exist sort of towards the bottom of an Sds-Page gel. But I want to take just a few minutes to explain how we form that gel. And so this is a polyacrylamide gel, and so it relies on the acrylamide compound here. And what we're going to do is see how this polymerization reaction occurs via radical chemistry. And there are two additional important reagents besides acrylamide, and that is APS, which is ammonium persulfate, has a formula S2O8 minus 2. And of course an ammonium counterion there. There's also the tetramethyl ethylene diamine, known as TMED compound, which looks like this. And these two reagents will react to form a series of radical compounds. And we're not going to worry about the exact chemistry and exactly what those radical compounds are, but just know in the end we make these, after the rearrangement, we make a radical that of course is very reactive because it's an unpaired electron. And that radical can react with acrylamide. So we've got the acrylamide here, and the radical will react with this terminal carbon like that, and we will get a bond cleavage then that occurs like this. And so what we end up with is a molecule with this radical group attached. I'm going to draw it on this particular acrylamide over here, and what was the carbon-carbon double bond would now be a single bond, and we have a radical now present on that carbon in acrylamide. And so we've made basically radical acrylamide at this point. And what can happen next is that radical acrylamide can react with another molecule of acrylamide, forming a new bond to it. And then we can break that carbon-carbon double bond, and we make another molecule of radical acrylamide that's already now attached to the previous molecule. And so then this radical acrylamide can react with another molecule of acrylamide, etc., hence polymerization, and we'll end up with a backbone of carbon atoms, CH-CH2, repeating like this. And if that's all we had, if we only had acrylamide, APS, and TMED, and of course buffer and other things like that present, we would just make a long linear polymer of polyacrylamide, and it would have a snotty texture, but it wouldn't be a gel. In order for it to be a gel, we need this compound, this bis-acrylamide shown here. Bis-acrylamide really is just two regular acrylamide molecules attached via this methylene linker here. And what that does now is, you can imagine, we can form a polymerization of acrylamide along these carbons and along these carbons, and now they are attached together, and we have now cross-linked the linear acrylamide. And so you can envision then we're making this sort of mesh like this with these cross-links, right? And this is, of course, in 3 dimensions. It's not just on a flat surface. And so we make this 3-dimensional gel matrix. Not only that, but we have all of these amide functionalities here, which are, of course, polar. So we've generated this polar 3-dimensional gel we're going to ask the proteins to migrate through because of this electric field that we put them in. So now we have the gel, let's think about the charges of these. And we should know by now that depending on the pH, proteins can be positive, negative, or neutral. And so how do we deal with that? And so we need a uniform charge. And to do this, what we're going to end up doing is exploiting the fact that proteins have a somewhat similar proportion of hydrophobic residues. And we're going to use the molecule SDS, this molecule right here. This is a negatively charged detergent, and it's going to bind to proteins in roughly about a 1 to 2 ratio, so 1 molecule of SDS per 2 amino acid residues. And it's going to mask any intrinsic charges that the protein has. And so even if that protein normally would be positively charged, because we're going to add so many SDS molecules to it, these are going to interact with those hydrophobic surface areas, that protein is going to have a uniformly negative charge to it overall. And so in general, proteins are going to have the same mass to charge ratio if they're treated with SDS. So that's good. They're all going to be negatively charged. They're all going to migrate the proper direction down in our Sds-Page gel rig that we'll see later on. In addition to uniform charge, we also need uniform shape, because you can imagine if I have proteins that are of equal molar mass, one of them is more spherical and one of them is, say, more hot dog shape, those certainly would migrate differently through a mesh. And so we need to unfold these proteins. They need to be completely denatured. And SDS also accomplishes that. It's a detergent. It's going to cause those proteins to denature. But there's also, remember, disulfide linkages that might be present in certain proteins. And so we need a reducing agent. And so dithiothreatol and beta-mercaptoethanol are 2 reducing agents that will convert those cysteine, those disulfide linkages, into just free sulfhydryl groups shown here that are no longer covalently attached. They also, these compounds are very stinky because they have those sulfurs in them. So we're always cautious about using those in the lab, because if you get them on your clothing, you're going to smell pretty bad yourself. So now what we've done, we've fully denatured the protein, and so they're all going to have a similar shape, this linear shape. They're all going to be negatively charged, and so now we're separating things that look like these two down here. We have small molecules that are these linear spaghetti molecules with SDS attached versus larger ones of a similar shape. Now, because of this, it's important to recognize that we cannot get any information about quaternary structure, right? Proteins fold up into form dimers and tetramers, etc., based on those non-covalent interactions. And so when we use SDS Page as an analytical technique, we are destroying any information about that. And so all we're really getting is the monomeric masses. We are not getting any information about whether a protein would form a dimer or a trimer, etc. There are things called native gels where we can get that kind of information. These do not use SDS, but they are very difficult to interpret because you can have different charges and they get kind of messy as a result of that. One other thing I want to talk about are the difference between resolving and stacking gels. And what I'm going to do is draw kind of a visual diagram of what one of our gels might look like. So we've got all these wells that are going to run our samples in. And remember, this way is up, and we're going to run the gels so the proteins migrate down. But actually, we're going to do 2 gels, one on top of the other. And so it's going to look kind of like this. And the bottom part is called the resolving gel, and this is where the proteins will actually separate. And this is, in a typical experiment, 10 to 12% acrylamide. Now the top part here is called the stacking gel, and this is usually something like 4% acrylamide. And so it's a much lower percentage. And the reason we do this is, let's say we load up our protein sample into one of these wells. we're going to apply that electric field, and then these samples are going to, it's going to start to migrate this way. It's entering this 4% relatively low density environment, and so it's kind of flying along. All the proteins are flying along. Maybe get a little bit of separation in there. But then those proteins are going to hit this interface where it jumps up to 10 to 12% acrylamide, and they're all going to kind of pile up, just like a car hitting a brick wall. They're all going to pile up, and they're going to start off with, enter the resolving gel, they're going to start migrating into that as a thin band as opposed to this blob like we have up here. And so in the end, we will get very, very thin, fine bands, which is certainly what we want to see. And so we don't always run stacking gels. Sometimes even gels you buy that are commercially produced might not have a stacking gel. They might just be only the resolving gel. But it will result in a more defined thin band versus one that might be a little more diffuse if there is no stacking gel. Now, once the gel has been run, we need a way to visualize where the proteins are, because they, of course, are going to be invisible. And there are a couple different ways to do that. Kumasi dye, shown down here, is the most common way. It has a certain detection limit where if you don't have enough protein, you won't really see anything. Silver staining then has a lower detection limit, and you can detect smaller amounts of protein. But most of the time, certainly what we're doing, Kumasi is going to be just fine for what we want. So this works the same way as the Bradford assay in some ways, where the protein and dye kind of interact with each other, and then it sort of permanently stays attached to the protein, and so we can see where the protein is within the gel. We also have to use a molecular weight marker, shown here, to determine what the size of our protein is. And so notice these are all values in kilodaltons, and so each of these bands here would represent a different protein standard that's added into this mixture of the molecular weight marker. And so that way we run this with our proteins, or in a lane beside our proteins, and we can see exactly, or estimate pretty effectively anyhow, what our molar mass of our protein is. And so this is what it should look like in the end. This is a Kumasi stained Sds-Page gel from a biochemistry research student, and you can see the lanes up here where the samples were loaded into. The gel was, of course, run and then stained with Kumasi and destained. And this lane on the far right, this is the molecular weight marker, and so you can see these different bands here that indicate the different sizes that we can use to estimate the size of our target protein. I don't have the legend for that on this slide, but you can certainly look it up, and we would be able to identify exactly what each of those bands represents. And on the far left, this is a crude sample, so a crude lysate, or a very impure protein, and then after the purification procedure has gone through, we would have this second lane there. And you can see then this target protein here, this is what we're after. It's the most dominant band throughout this whole thing. And certainly in the crude there were lots of contaminants that were present, but in this more pure sample we really only just have this one minor contaminant up here, and we've gotten rid of a lot of those other things. And if we know what the molecular weight of this standard rate here is, we can get a pretty good idea what the molecular weight of our protein is, just to confirm that that's what we have, and the protein's been expressed in full length and hasn't been degraded by proteases or anything like that. So Sds-Page is a very common technique in biochemistry to look at purity of a protein, and we're going to definitely utilize this.